This is Jose Gonzalez. I've been with Illumina for a while, and today's topic is GenomeStudio Advanced Analysis Tools. Today our objectives are to describe a number of tools and features in GenomeStudio which are not widely known, how they work and what they do, and how you may want to use them. Our purpose is to empower users to use the software to its full capabilities. So let's go over some of the tools we will be reviewing. First, we'll be looking at how to import columns. This is a tool that is available in all GenomeStudio modules, and we're going to go over how this tool can be used, for example, adding annotations and perhaps marking particular markers for filtering. All right. In addition, we're going to do some review of tools that are specific to genotyping and specific to methylation and gene expression. In genotyping, we'll be looking at the GenderEst tool, the Reproducibility and Heritability tool, the Concordance tool, and some cluster editing tools. In the methylation and gene expression, we'll be looking at scatter plots, and if we have time, we'll go over how to use SNP-targeted assays in the GenomeStudio project as identification tool. So, without further ado, let's start with importing columns. Columns can be imported into any table in the GenomeStudio software. In the samples table, in the full data table, or the SNP table in genotyping, and the sample methylation profile or group methylation profile in the methylation module. So all tables have the import columns button in the button row. As you can see here, it looks like a piece of paper with an arrow pointing into it. Now, to import columns, the first thing you need to do is generate an import table. An import table is a tab-delimited text file, and it has two or more columns. The first column is what we call the key column. It references existing data in the table to which you will be importing. So the key column heading must exactly match the heading of the corresponding column that you're keying from in GenomeStudio. The remaining columns are the columns that you will be importing into the project. And the column headings for these can be any text. There's no limitation for that. So let's take a look at a simple example here. I've created an import table using gender as the key column. And you can see here in the table I have values female, male, unknown. And the column I'm importing is called Hawaiian. And in that column are values that correspond to the translations for man and woman. All right, and we will be importing this into the samples table of a GenomeStudio project. Now, the next step after creating this tab-delimited text column is to click on the Import Columns button. Now, in the dialog box that appears, you click on the Browse button and browse to the import table and click OK. Now, what will result, the column Hawaiian has been added to the far left of the table. Any column that you add will initially begin at the far left once it's been added. You can move that fairly easily by clicking the heading and dragging it to any portion of the table. The values that have been added are based on matching values in the key column. Non-matching values, for example, the unknown, which doesn't exist in the gender column, are ignored. Also note that for repeated values in the key column, only the first value is referenced. As you see here, female is listed twice under the gender column of the table. However, only the first female is considered, and only the first value is imported into the column. The second value for female is ignored. Now, key column values are case sensitive, so if I had put female with a small letter, it would not have been imported at all. All right, let's take a look at an example application. Let's say that you've created a GenomeStudio project but at the time that you created the project, you did not have phenotype data, so you did not include the phenotype data as additional columns in the samples table. So now you've actually obtained that information, and you want to add it to the samples table. Is there a way to do it without having to recreate the project using a sample table, a samples file with the new information added? And the answer, of course, is yes. You can import this without having to recreate the project. In this example, we have a import table in which we reference the sample ID column, and then we add the column phenotype with the values corresponding to diabetic or normal in this particular case. When you import this table in, you see here in the samples table that the phenotype column that you imported has been added to the far left, and corresponding values that match to the sample ID have been added to the samples table. Let's look at another example application. Let's say you have a list of 2,000 genes of interest that are a subset of the 48,000 markers that are on the gene expression HT12 project that you have. And you want to explain only 2,000 of those markers because those are the genes of interest for your particular project. Is there a way to select those in bulk instead of having to select them one by one and then showing them? And of course, the answer is yes. You can use the import columns function to import a marker column that highlights only the genes you're interested in, and then use the GenomeStudio filter tool to filter out all the probes except those that are highlighted by the markers. So how do we do that? We'll create an import table. In this case, we're not looking at 2,000. I've only put a handful of genes that we're interested in. And so on the left side, I have used the symbol column as the key column on the left side. The next column, which I've simply called marker, you can call it anything you want, contains the value number 1, or any valid character of your choosing you can put in there. In fact, you can put different values in there if you wish to use it as a sorting tool to prioritize which symbols you're most interested in. So now we've created a import table here and saved it as a tab delimited text file. We now import this into the GenomeStudio project, into the sample probe profile of the project. And what you see here is that when you import this, again, the marker column is added to the far left. Now, in the GenomeStudio Gene Expression product, there are often multiple probes that target the same gene. So you can see here that there are 3 probe IDs or more for this gene A2BP1. Now, what you see is that the A2BP1, which is listed only once in the column import, is actually marked for all of the probes that have that particular symbol. So you can see that all qualifying key values will be marked with the marker. All right. Now, the next step, of course, is to use the marker information as a handle for sorting and filtering. You can select the marker column by clicking on the column header, and then click on the button Sort Column Descending to bring all the rows marked number 1 to the top of the table. You can then select these rows, right-click in the table, and select Show Only Selected Rows from the pop-up menu in order to display only the marked rows. Alternatively, you could click in the Filter tool, and if you click on the Filter tool, which is that button that's shaped like a funnel, you can select the value marker for column, and as you notice, any column that you add will now be visible as a column, and you can say marker equals 1 and click on the arrow here to add this to the filter tree. Once you click OK, again, the result will be that only the values which have the marker value of 1 will be displayed in the table. All right? So, to summarize, importing columns is available in all GenomeStudio tables. Import table is a tab delimited text file. the first column is the key column, and it references an existing column in the table, and the column header for the key column must match the header in the existing column in the GenomeStudio table. Mismatches and repeats in the key column are ignored, and all repeat values in the target table will receive entries. And the column import is useful for adding annotations to the sample table after creating the project, and for marking large subsets of data rows for sorting and filtering. A few caveats. Number one, you cannot use the import column to modify existing information in standard columns. So you cannot modify the standard columns that are already part of the GenomeStudio table. You can only modify and add columns to the existing columns. Columns imported cannot have the same name as an existing column. And finally, added columns are removable. In any column that you've added, you can right-click at the header, and the option to remove the column will show. All right. Now, let's move on to some of the tools that you can see in the genotyping module. And so we will be looking at GenderEst, reproducibility and heritability, concordance, and cluster editing. GenderEst is a tool that uses data from the X and Y chromosomes of each sample to determine sample gender. If the sample genders are unknown, you can use GenderEst to calculate the genders for each sample and populate the gender column. If the sample genders are known, you can run GenderEst and not overwrite the gender values, and then compare the values that you get from GenderEst to the values that you've entered in the gender column. All right? Now, there is a new version of GenderEst, and so there are two versions available. You can choose which one you wish to use. In the original version of GenderEst, the GenderEst algorithm collects all the SNPs on the X locus, counts all the heterozygous or AB genotypes on the subset. If there are less than 20 total SNPs on the X chromosome, then the gender is registered as unknown. If there are greater than 20 SNPs on the X chromosome, if the percent of AB is 10% or more, then the algorithm calls the gender female. For greater than 20 SNPs on the X, if the percent is less than 10%, then it calls the gender male. And depending on the coverage, for example, SNPs on the X chromosome, that is to say, SNPs on the X chromosome, the gender estimation accuracy will vary. What I mean by this is that there are some arrays which contain only rare variants. So even for X chromosome on female, where you would hope to see some heterozygous calls, if all of the markers are rare variants, then you're going to get a low heterozygosity regardless of the gender. And so this algorithm would not work. For example, any exome-based arrays from Illumina will have very rare variants. Now, GenderEst algorithm has been updated, and instead of relying on heterozygosity and genotypes, it relies on intensities. So for version 2 of GenderEst, the algorithm counts all autosomal loci and Y loci. If the autosomal loci are below 100 or the Y loci are below 20, then it defaults to the older GenderEst algorithm. If the call rate of the autosomal loci is below 97%, then it calls the gender unknown. We want to have reliable data, and so if the call rates are low, then we cannot rely on the data, so it calls the gender as unknown. If the median Y density, that is normalized Y intensity, is greater than 0.3, then it will call the sample as a male. If it does not meet this criterion, then it will move on to the next algorithm criterion. If the median X intensity is less than 0.9, then we will call it unknown. Otherwise, it will call it a female. All right? So those are the two GenderEst algorithms. You can modify the GenderEst thresholds in the GenomeStudio config file. It's in the GenomeStudio executable program directory, and you can manually select between version 1 and version 2 of GenderEst and modify the parameters for calling the gender. We recommend contacting tech support for assistance when you modify this config file. All right, just a quick look at the data that comes from male and female chromosomes. You can see at the top we have a male sample, and the X chromosome lacks any heterozygosity, whereas the female X chromosome shows heterozygosity because it has 2 X's. On the Y chromosome, the male, it shows definite A and B genotypes with a small amount of heterozygosity due to the pseudoautosomal regions that are present. And then the Y chromosome on a female should yield extremely low, if any, intensities. And if you look at the B allele frequency values, it'll look like a waterfall, simply because there's no actual ratio between genotypes because the intensities are very low. So those are the differences that algorithm 1 tends to capitalize on, whereas algorithm 2 will look at overall normalized intensities. Now, the GenderEst column in the samples table is by default not visible. In order to make it visible, you have to click on the column chooser. The column chooser is a button on the top of the rows, and all tables have this button. It essentially has 3 vertical blue bars on it, and once you click it, you get this dialog box, and you can see that the GenderEst is under the hidden columns. You can simply click and drag it to the show columns, and you can place it by clicking and dragging in between any of the visible columns you want. If you click on the show button, it'll place the GenderEst at the very end of this list, and that will put it at the very far right of the table. So clicking and dragging it allows you to place it where you wish. All right. To run GenderEst, you go to the samples table and select all the samples for which you wish to estimate gender. Right-click on the table, and in the pop-up menu that appears, select Estimate Gender for selected samples. GenomeStudio will ask you, would you like to populate the gender column with the results of the calculation? Now, if you choose yes, then GenderEst will overwrite any genders that are already in the gender column. So if you've already mapped your samples, you may want to say no to this and run the GenderEst so that you can compare the results of the GenderEst algorithm to what you have entered and confirm your mapping of your samples or confirm the genders of your samples. All right? Now, once you've made your selection, GenderEst will calculate the gender and simply populate the columns as designated. So to summarize, the original version uses X chromosome genotypes to calculate sample gender, whereas the version 2 uses X and Y chromosome intensities, which are a little more reliable in terms of determining gender. There is an option to populate the gender column if you wish, and it can be applied to determine your sample gender if you don't know it, or if you know it, you can use it to confirm your sample mapping in the project. All right. Moving on, let's look at the reproducibility and heritability tools in GenomeStudio. So, the reproducibility tool allows you to designate replicate samples in the GenomeStudio Genotyping project. It calculates replicate error rates and highlights assays which show discrepant genotypes between the replicates. The heritability tool does something similar. It allows you to designate parent and child relationships within the GenomeStudio project, and then it detects assays which show Mendelian heritability discrepancies between parent and child designated samples and calculates error rates for those associated samples. It will also highlight those assays which show discrepant genotypes between the parent and child. Now, for both of these tools, there are 2 ways to designate replicates or assign parent-child relationships. You can designate them in the sample sheet before you create the project, or after creating the project, if you haven't designated them in the sample sheet, you can manually designate them in the analysis menu. All right, let's take a look at a sample sheet and how you would designate replicates. Here you see a standard sample sheet for a genotyping project. we have the sample ID column, the centrix IDs and positions for those columns, and then the right side you can see here the column for replicates. And what you can do is designate, if this is the first sample in the project and you have a replicate for it, you can type in the replicate ID here. And this should actually read NA18951-R. You can actually, after looking at this, the first sample is the R version, and then the sample replicate is the one without the R in this case. So you only need to make this designation once. In other words, you don't have to go to the entry of the second one and repeat the designation by naming the alternative sample in the column. So just do it once, all right? And likewise, you can see in the right side, there are columns for parent 1 and parent 2. So if you have a sample which is a child, you can designate which samples are parent 1 and parent 2. So for manual designation, if you did not do this when you created the project, you have the options under the analysis menu to edit replicates or edit parental relationships. All right. To edit replicates, select that option in the menu, and you will see two columns. Both columns will list all of the samples in the project. You simply click on the first of the replicate samples on the left, and then the other of the replicate samples on the right, and click on Add, and that pair will appear in the table on the right side. There's also a checkbox next to the pairs, and this allows you to select or deselect the replicates for analysis. All right. Now, once these have all been designated, you can click on OK. Similarly, for heritability, you can manually edit parent relationships. You can select parent 1, parent 2, and child in the drop-down menus and click on Add and click on OK when you finish. And GenomeStudio will start to calculate the heritability and update the SNP statistics. Note that the order of the parents and the child in the table, it's important because you may make assumptions of what comes first, but you have to pay attention to which column is which. Now, after designating these replicates to initiate the reproducibility and heritability to the calculations, you need to click on this menu item under Analysis, select Update Heritability and Reproducibility Errors from the Analysis menu. Now, once those have been updated, what you'll notice is that in the samples table, several of the columns will turn red. This is because the samples table is not automatically updated when you make changes to the statistics in the SNP table or the full data table. Those must be manually initiated. What you will see is when you make any changes in the SNP table or the full data table, a button on the far left will appear in the form of a calculator. You'll need to click on that calculator in order to clear these red columns and fill in the appropriate values. All right, so again, once you've updated the heritability and the reproducibility information, you need to update manually the information in the samples table. Now, once you've actually clicked on the calculator and it's completed its calculations, you'll see that the PC errors and PPC errors and the rep errors columns have been populated with values. This will give you a count of the number of assays that show contradictions in the assumptions of parent-child relationship or replicate. So in this particular example, you can see in the SNP table, this particular SNP has one parent-parent-child error and one rep error. So for each SNP assay, the SNP table will list the number of discrepant replicate pairs and parent-parent-child errors. You'll need to click on the heading of the column of interest and sort Z to A to bring the assays with the highest numbers of errors to the top. All right. So now, once you've actually determined which of the SNP assays are showing these conflicts, you can actually see what the conflicts are visually. You can click on any line in the SNP table which contains reproducibility or parent-parent-child errors, and what you'll see below that there's a table called the errors table, and this table is visible by default, and it's blank initially. But once you click on a SNP that shows an error, the error will be listed below in the errors table. So for this highlighted SNP, there is a rep error at this particular SNP, and you can see the values for the sample. This is the sample identification for the child, or for the rep 1. And then here's rep 2. So the two replicate errors that are discrepant are between this sample and that sample. You can see the genotypes listed. So one sample has BB and the other has AA. So clearly there is an error in the replicate for this particular SNP. Below, there are also additional errors for the SNP. There are parent-parent-child conflicts between this sample and this sample and this sample. You can see the two parents and the child, their particular genotypes. And you can see, for example, that in this case, the child has an AA genotype and the parent has a BB genotype, which is not possible. So the type of errors, along with the sample IDs and the discrepant genotypes, are displayed in the errors table. In addition, you can graphically observe these particular conflicts. If you click on the particular error in the errors table, the 2 or 3 samples that are involved will highlight in the samples table, and in the SNP graph, you will see the data point for those particular samples. So you can confirm visually whether you agree with the assessment that there's a replicate error or a parent-parent-child error by visually inspecting the data points for those samples. All right. And you can see in this case, for this replicate error, the data points indeed are far apart. One is in the BB cluster and one is in the AA cluster, and there can be no questioning about the conflict. All right. So how can this be used? Well, you can confirm sample mapping in your project. If you sprinkle a few replicate samples throughout a large project and then you calculate the replicate errors, you can find out if your sample mapping has been done correctly. If it was done correctly, you would expect low replicate or parent-child errors, and your sample mapping is likely correct. If their error rates, on the other hand, are high, then you may need to inspect how you map your samples in your sample sheet. It may be incorrect, and you may have to correct it. You can confirm the overall quality of the project as well. Low error rates indicate that the data quality is high. And also you can view discrepant data points graphically in the SNP graph to determine the reliability of the genotype calls for that particular SNP. You could look at the clustering and see how well the algorithm was able to cluster the data. You may modify the clusters using the tools that we will cover with regard to editing SNPs. All right. So in summary, reproducibility and heritability tool allows you to designate replicate samples and parent-parent-child relationships via either the sample sheet or by editing them manually after the project has been created. It allows you to view and display and review discrepant replicate and parent-parent-child genotypes via the SNP table, the errors table, and the SNP graph. And it will allow you to review reproducibility and heritability data to confirm your sample identities and assess data quality. All right. The next tool we will be looking at is Concordance. Now, Concordance allows you to import genotype calls from the samples of one GenomeStudio project into another GenomeStudio project and calculate concordance between genotype calls for samples in between the two projects which overlap. So essentially, it allows you to compare genotype calls from one project and another project. And it is very similar to including replicate samples, but one of the differences is that the Concordance tool is not limited to bead chips of the exact same type. So when you import a replicate sample into a project, it must have been run on the same type of chip in order for the replicate calculations to be valid. Not so for Concordance. If, for example, you run a GenomeStudio project with a 5 million SNP chip, and you run a subsequent project with a 2.5 million SNP chip that's a subset of the 5 million, you can analyze the genotype calls, import them from one into the other to compare the genotypes. And it does not require that they come from the same type of chip. It only requires that they have some overlap in the markers. Now, the concordance tool is not useful for CNV analysis because it only transfers genotype calls. It does not import statistics such as B allele frequency, log-R ratio. Those are not imported when you import sample genotype calls for concordance analysis. It answers the question, am I getting the same genotype calls from one project as another project for the same sample, as opposed to sample replicates, which answers the question, am I getting the same genotype calls and B allele frequency log-R ratio from one sample versus the same sample within the same project using the same type of chip? Now here's the suggested workflow. Run a gold standard genotyping project which contains the control samples that you intend to use in subsequent genotyping projects. Edit the clusters in that project and optimize the genotype calls so that you have complete confidence in those genotype calls. You can then export the genotypes using the concordance tool, and then in subsequent genotyping projects, run some of these gold standard samples as controls on the subsequent genotyping projects. Import these genotypes from the previously exported gold standard into the current project and run the concordance. This will give you essentially the answer to, did I get the same genotypes? And it's a measure of QC to see if you're clustering, to see if the analysis and see if the quality of your data is the same as previous. And it allows you also to edit clusters as needed in order to allow this to conform to your previous gold standard results. So how do you execute concordance? First, you create your genotyping project with your gold standard samples, and then export those calls from the project. Under the analysis menu, you can export the allele calls. It's down there on the menu. And after that, you'll get a dialog box, and you will need to choose the format in which you export the genotypes. You will either choose from the top-bottom strand, which allows you to export the top strand alleles, or the plus-minus strand option, which exports the plus strand or reference strand alleles. If you have a reference strand, I would suggest using that because that's the accepted standard for today. You need to keep track of which option you choose when you do the export because you'll need to import them into the subsequent GenomeStudio project using that same standard. So next, you'll browse and create a new folder to put these export files in and select a name that will allow you to keep track of this folder very easily. And then once you export it, these files will be created in the directory. And you'll see here that the files are named after, these binary files are named after the samples that you exported. So the directory will contain binary files for each sample that you exported. It will also include an index file which lists the SNPs that were exported, the names of the SNPs that were in the project that were exported. There's a binary version and a text version which you can open in Excel or Notepad. And then there's a nice little file called metadata, which actually reminds you which option you chose for the reference option. So if you ever forget, you can just open the metadata file and it will simply tell you. All right? The next step is to run your second or third or subsequent GenomeStudio projects and then import the genotype calls for those control samples into your project. On the analysis menu, select Import Allele Calls. You will then have a dialog box and you will be asked to navigate to those calls. Simply navigate to the directory that you saved those binary files in, and then make sure you select the appropriate top bot or plus-minus strand option, and it will show you the names of the files listed in there. Hopefully the files that you exported will overlap with the files that are in your project, at least a few. Click on OK, and you will have imported the genotype calls. After the concordance calls calculations are finished, the data is not displayed by default. In the samples table, click on the column chooser and add the column concordance. So in the samples table, you will have the concordance calculated only after you click on the calculator button. So you've added the column for concordance, but there have been no calculations made yet. So let's click on the calculator button. And, I believe I'm missing a slide here. But what will happen is you will see in the concordance a percentage call of the percent of concordance that that sample has if there are concordance values imported into that. Now, in order to view the actual concordance data for each individual SNP, you'll need again to go to the column chooser in the full data table. Now in the full data table, you will need to add 3 subcolumns: the top alleles, the import calls, and the concordance. So you'll need to select those and drag them over to the displayed columns. Now once those are imported, what you will see are those 3 columns in the subcolumns for each sample. So in addition to the genotype call for each sample, you'll have the top alleles imported for each sample, the import calls for each sample, and then whether the call between the sample and the import was concordant. So this is another difference between adding a replicate sample and adding a concordance. In replicate samples, you're actually including an additional sample in the project, whereas with concordance, you're importing genotypes into an existing sample in the project. So you can see here, essentially you're comparing what you've imported to what the project has called. And if they are concordant, if they match, then the value in the concordance will be 1. If it's a SNP that was not overlapping and it wasn't analyzed, then you'll see a -1. And if there's a mismatch, you'll see a 0 in the concordance column. You can then sort by concordance and analyze those which have mismatches to see if you can explain or figure out why those are not concordant. So in summary, concordance allows you to import genotype calls from a previous project into current projects to check for reproducibility. Only overlapping samples with exactly the same sample names will have genotypes imported for the comparison. Different from establishing replicate samples, it's different because data can be imported from projects based on different bead chip types. Only overlapping SNP assays will be imported. It's useful for confirming quality of current data in comparison to past projects. It's also useful for confirming sample mapping, much like replicate samples. All right, let's move on to our next topic, cluster editing. Now, there are 2 tools I want to discuss, the F2 function key and the cluster reshaping tool. The F2 function key is useful if you're running through a long list of SNP assays in your panel which are underperforming and which you'd like to remove from the analysis. So it allows you to quickly review visually and remove from analysis a long series of assays. The cluster reshaping tool allows you to edit how the clusters are designated, allows you to edit the size and shape and position of the cluster ovals. So there is a recorded webinar, GenomeStudio Genotyping, Creating Custom Cluster Files for Infinium Analysis. This explains why custom cluster files are created and suggests methods for sorting and filtering and identifying SNPs that require attention and cluster editing or removal. And SNP assays which underperform in a GenomeStudio project across the board can be zeroed so that the results are excluded from all calculations. Now, you may wish to zero a SNP or exclude it from analysis if, for example, there are none or few data points were derived from the samples for assay, or if this data do not form distinct clusters that you can trust the genotype assignments. Now, in order to zero a SNP, normally you go 1 by 1. So you will select the SNP in the SNP table or the full data table, right-click, and in the pop-up menu that appears, you will select the option to zero the selected SNP. So that is a manual process. Now, F2 is an alternate way to quickly zero a SNP. If you have sorted your SNPs in the GenomeStudio by a particular parameter, for example, the number of calls, and you have a long series of SNPs that will need to be zeroed, but want to review each one before zeroing, you can be the F2 key can be very helpful. Many people will simply highlight the whole list of SNPs that they think need to be zeroed, right-click and say zero selected SNPs, but there are those SNPs that happen to perform poorly, but you may want to actually have the opportunity to review before you zero. And so if you've sorted your samples, your SNPs rather, into an order where you have the questionable SNPs at the top, when you have hit a highlighted SNP that you intend to zero, simply hit the F2 button. That will zero the highlighted SNP and automatically advance the cursor to the next SNP in the list, instead of having to right-click with the mouse and select the item from the pop-up list. So if you're editing a large chip data set and you have a lot of SNPs that you need to go through manually, the F2 can get you through a lot of them very quickly. All right? The cluster reshaping tool, keep in mind that the GenTrain algorithm, which analyzes the data and forms clusters in the data, is designed to mimic how a user would manually cluster the data from SNP assays and assign genotypes to the data point in an Infinium genotyping array. However, it's not infallible. And there may be instances where the clustering algorithm places the clusters in locations which you disagree with. And you visually can see that you could do a better job. So the application of a cluster file also is a shortcut that eliminates GenTrain clustering, but the positions applied from the cluster file may be slightly different from your current project. And that is because the positions of the clusters on the cluster file may be affected by local conditions in the lab that created the cluster file. If the lab ambient temperature is extremely cold, for example, the cluster positions in that project may be affected compared to the cluster positions in your project. So cluster reshaping and editing is a way to fine-tune even a cluster file that you've used. So you may sort your data after applying a cluster file, looking for instances where the clustering of the project doesn't fit the clustering from the cluster file. And this tool will allow you to modify those clusters. So I just said that allows you to fine-tune and edit the positions of the clusters. To reposition a cluster, to actually change the position of the cluster, move your cursor to the centroid. That's the plus-shaped mark at the center of the oval. Hold the Shift key down, and then click and drag that centroid in order to reposition the oval. All right, in this example, we're simply clicking and dragging the center of that oval around the AA cluster with the 32 samples and moving it down by clicking and dragging it. In addition, you have the opportunity to change the size and shape of the ovals. Simply move your cursor to the edge of the oval and it will again change shape, and then click and drag the edge of that oval in order to move the edge of the oval and reshape it. All right. So let's take a look at scatter plot tools. Now the example I'm going to give is a gene expression project, but the same applies to the GenomeStudio Methylation module. And we're going to look at scatter plots, and if we have time, we'll look at the SNP targeted markers. We are running a little bit longer than I expected, but let's take a look at the scatter plots. Scatter plots are available in all GenomeStudio tables. They have applications for methylation and gene expression. The plot any two columns in the table against each other, and it calculates a correlation coefficient. Now in this example, we're going to correlate intensity data between two replicate samples. In the sample probe profile, click on the scatter plot tool. Select the sample for the first sample, and then the subcolumn we're going to use average signal for that sample, and click on the y-axis button. Similarly, we're going to click on the x-axis button for another sample and its average signal as well. And click on OK. And you will derive a scatter plot from these two data. And you can see here, in this particular case, the correlation between the two samples is very high. The R-squared is 0.9944. Non-replicate samples tend to show more scatter and lower correlation efficiency. You can see here the correlation efficiency is 96%, and the data is more scattered. Now sometimes the scatter plot can be useful not only for comparing replicate samples and seeing how well they correlate, but actually for identifying markers which don't correlate between two different samples or two different groups. For example here, let's look at the identification of differentially expressed genes. In the group probe profile of a gene expression project, you can select the scatter plot tool and assign one group to the y-axis and one group to the x-axis. But you can see here again is a larger scatter that you wouldn't expect to see in samples that are replicates. In the scatter plot, click on the N-fold button, which is right there. And what will happen is 3 lines will appear. The diagonal line that runs from corner to corner is the identity line. This is a line that represents data points that replicate each other, that fall on the axis at the same point. The two-fold lines represent where the data point would fall if they were two-fold different. For example, one two-fold greater than the other. And so data points outside the two-fold lines show a greater than two-fold differential in intensities. What's nice about this is you can select any of these regions, whether they're within two-fold or outside of two-fold. So you can select fold change greater than 2 and say select. And those samples which show greater than two-fold difference will be highlighted in blue. All right, you can do the same for any of the other regions. You can select less than two-fold and so forth. Now in the group probe profile, what results is that the highlighted probes in the graph are also highlighted in the group probe profile table, which is nice because now you can right-click on the table and say show only selected rows, and you'll be viewing the probes which show a two-fold great difference. All right? And you can be repeated for any of these 4 regions divided by the identity lines and the fold lines. And the fold lines can also be adjusted by adjusting the number value in the area where it says n-fold. And you can click on the up or down arrow keys or highlight it and type in your value. Now the axis scales also can be changed from linear to logarithmic by clicking on the drop-down arrow next to linear. What's nice about this is it brings by scaling it to logarithmic, it brings the two-fold arrow lines parallel to the identity line, and it stretches out the data. The data is still the same, it's just viewed with the parallel lines instead of the converging lines. All right. Scatter plots allow you to visualize how well two samples or groups are correlating. It also allows you to select differentially expressed or differentially methylated targets between pairs of samples or groups. Okay. I'm going to skip this section on the methylation sample identity using scatter plots, and let's go on to online resources here. There's some general training and webinar recordings listed at this site, and also some support pages for the software. This is the software, the link to the GenomeStudio software and documentation, and some GenomeStudio specific training videos and webinars. All right, and I'd like to open this up now to any questions that you might have